So welcome everybody for our really very special online virtual ESID meeting that is a little bit strange for us because I cannot see you, you cannot see me, but anyway, this is the situation now. We are starting with the session which is called Clinico-Pathological Discussion, and the real topic is primary immune deficiencies and the nervous system. I am chairing the session together with Neves, who will guide you through the end of the session with questions and answers and discussion. So, concerning the topic, I find it very cool, and once I heard that we are chairing specifically this session, I went to check what's new about the connection between immune and the nervous system, and I did find quite nice picture which I am sharing with you that shows multiple interconnection between these two systems. And of course, immune system is an integral part of central nervous system and also interact with peripheral nervous system. And what's rather new, that gut immune system and microbiota also interact with central nervous system, but also with peripheral nerves. Concerning our specific topic, which is primary immune deficiencies and nervous system, it's also quite interesting because you might have infectious and non-infectious manifestations in CNS. And once I thought from my practice with immune deficient patient, I realized that in the history we did not see so that many neurological complications. I remember some XLA patients, for example, and in the history they had enteroviral meningoencephalitis, which we do not see anymore. We also have some special XLA patients who have big deletion in the X chromosome, and it encompasses a series of other genes. They are responsible for the quite severe neurological impairment. But it was in the past, and now in the present, the picture is completely different. We have so many new immunodeficiencies and new categories of primary immune deficiencies, and in many of them we see some neurological manifestation of the immune deficiency. I listed here several categories of immune deficiencies which are connected. I know some patients from these categories connected with neurological impairment, but there might be many others. And I am really looking forward to our session. It is listed here. It's on Thursday in the afternoon, 14:15, and I am really looking forward to your contribution and to news about the PIDs and the nervous system. So welcome all.
Well, thank you very much indeed for the invitation to give a talk today for the European Society for Immunodeficiencies, your excellent online forum. These are strange times for everyone, but I hope that you can see and understand my presentation. So I've been asked to give a talk about a fairly broad subject, which is immunity in the brain. And just to start, I have some disclosures here. So I have nothing to disclose. I have no relevant disclosures to the content of the talk. My funding comes from the Wellcome Trust and the Medical Research Council, and the only interaction I have with industry is as a scientific advisor to the regulators, to the MHRA, and to the EMA. So just to give a little bit about my background, I'm not a pediatrician or a geneticist or work primarily in the immunodeficiency field. I'm an adult neurologist and I practice in Edinburgh. This is a very beautiful clinic that's recently been built with a generous donation from the author J.K. Rowling to build a clinic in memory of her mother, who had MS. And we do clinics there for adults with neuroinflammatory disease, but also I have a group within the UK Dementia Research Institute, which is a UK-wide Research Institute to try and understand the contribution of particularly neuroinflammation to brain disease as a whole. So I was going to talk in broad terms, first of all, about immunity and the brain, and just to give a very broad overview of what is special, or maybe what is not so special about the brain and the immune system. But I want to have a more focused look at a pathway which I think is very interesting, which links up fields of immunodeficiency, links up fields of autoinflammation, and also links up with complex autoimmune diseases, genetically complex autoimmune diseases of adulthood as well. And that's the type 1 interferon response. So we're going to start broad and then go a little bit deeper. So just to start, it's an important question to think, well, what does make the brain special? Well, actually, as an organ, it's not that much different from the other organs, and that a lot of immunology is still dictated and influenced by the peripheral immune system, and that is just as important in neuroinflammatory disorders as the brain-resident immune cells. And so understanding peripheral immunity and peripheral immune cells is really critical. It has been in, for example, the field of multiple sclerosis, really the key to understanding and treating that disease. But there are differences, most notably, and probably most well known, being that of immune barriers that cause a degree, but not so much, not a huge extent, but some degree of immune privilege within the brain. And those immune barriers include, for example, the blood-brain barrier, which limits access to the central nervous system. The blood-brain barrier is formed by tight junctions from endothelial cells, but also it's a more complex structure than that. There are also contributions from neural cells and also from astrocytic end feet. And it's not just the blood-brain barrier that acts as an immune barrier. Here on this diagram, we have the blood-brain barrier, but also in the bottom right-hand corner, the blood-CSF barrier. And there are important immune trafficking mechanisms in both of those. But also things that are particularly specific to the brain are brain-resident immune cells. And these can include, for example, perivascular macrophages. But also a lot of attention has focused in the last few years of the role of microglia in health and disease. And those are both in primary neuroinflammatory disorders, but also in diseases such as Alzheimer's disease, where it's increasingly recognized that microglia, as the resident immune cell of the brain, the sort of professional resident immune cell of the brain, play an increasingly important role. And this is a review, this picture is taken from a review from Marco Prince and Joseph Priller, who have published some really fabulous work, particularly over the last couple of years, on human microglia, and our understanding of that is really accelerating. But also, we increasingly understand that neuroglia themselves, the neuronal cells, the astrocytes, oligodendrocytes, have innate immune mechanisms. And so you don't have to be, as it were, a professional immune cell to have an immune response within the brain. And there is quite a lot of complex neurobiology that links up the immune system and the brain, an example of which is the influence of microglia on synaptic pruning, on refining the connections between neurons, and the phagocytosis of synapses that can occur as part of their homeostatic mechanisms. So this is a really very rich area. It's an area that's traditionally, of course, been very difficult to understand because in human neuroinflammatory disease, because of the remoteness of the brain. I often feel very jealous of dermatologists and renal physicians who have much easier access, not necessarily easier access, but have closer access to studying the pathological events through either skin biopsy or renal biopsy. And it's very much more difficult to do that in primary CNS disease. And as a consequence, quite a lot of this is still being unravelled. And in fact, that was one of the reasons why I've always been very interested in the type 1 interferon response, because it seems a very tractable way to try and study and understand brain health and disease. And I'm going to talk briefly about the role in viral infection. I'm very aware that there are people who are much more qualified than myself to talk about that, speaking afterwards. So I will I hope, whet your appetite for some of the talks that are to come on that. But also to talk about how interferon can cause brain disease, and also the nuanced role that it has in adult neuroinflammatory disease as well. But also the insights that these different disciplines can sometimes have on each other, and how we can, by studying adult neuroinflammatory disease together with, for example, type 1 interferonopathies or viral infections, we can get a better synthesis of what's going on within the brain. So this is Alex Isaacs and his classic paper with John Lindemann from 1957 about the antiviral interferon interfering substance. And when we talk about type 1 interferon, of course, this field is getting more and more complex, but often what is referred to are the many type 1, the multiple interferon alpha subtypes and the single interferon beta. But of course, that is signaling through the type 1 interferon receptor with its two subunits, which then leads to a broad and complex downstream transcriptional response, and particularly of interferon response genes. And that's what the cartoon I've shown there is, of course, an oversimplification, but I think it's useful to look at this in a fairly simplistic way. Type 1 interferons, of course, are hugely pleiotropic cytokines. They play a role in many different aspects of immunity, cancer biology, and other aspects of biology. But if we look at this in a very simplistic way, the type 1 interferon response being a predominant form of host defense against viral infection of the brain, we can then classify in a rather simplistic way the problems of the interferon response being ones of underactivity, where type 1 interferon does either not respond appropriately at any stage of its pathway, which can then predispose to viral infections of the brain. But also we have a look at when type 1 interferon response can be overactive, as it were, trigger happy, and can cause neuroinflammation. And we can perhaps look at these as extreme phenotypes, and this is a classic paper now from Ion Gresser and Jean-Laurent Casanova, looking at herpes simplex encephalitis and the biological basis of that in human STAT1 deficiency and impaired response to interferon alpha and beta. And I'm sure after this there will be many more talks on the deficiencies of this pathway in primary immunodeficiencies. So I won't dwell on that. I would be keen to talk about the opposite of that, which is really to have insights that we get from neuroinflammation about an overactive interferon response. And these are, in terms of autoinflammation, increasingly referred to as type 1 interferonopathies. And this is a field that has really been pioneered by Yannick Crowe and others. I've been very fortunate in Edinburgh to work with Yannick and also with Andrew Jackson, who have really defined many aspects of this field in terms of both its clinical application and biology. And the first detailed description of this was in the early 1980s from Jean Cardi and François Gutierrez, who described a genetic mimic of congenital viral brain infection. where neonates presented with encephalitis, and specific clinical features of raised CSF interferon alpha, severe and diffuse brain disease, intracranial calcification. And the unraveling of the genetic basis of this has been an extremely rich exploration of the type 1 interferon pathway and how it functions and malfunctions in humans. The first mutations in terms of nucleases and ribonucleases that were identified in both TREX1 and the three components of the ribonuclease H2 complex give insight into aberrant nucleic acid processing and the potential role of immunostimulatory nucleic acids in terms of stimulating stimulating viral sensors, but also with subsequent discovery of other enzymes involved in nucleic acid processing, RNA editing, but also gain of function mutations in nucleic acid sensors, have led really, I think, to a much richer and deep understanding of the spectrum of innate immune activation that is seen, that although largely affects the brain, can also affect other organs, particularly the skin, but also in other conditions such as can affect the lung and guts. And also there are very specific patterns of neurological disease in some types of interferonopathy, most classically perhaps with bilateral striatal necrosis with ADAR1 mutations. And this is a fabulous review from Matteo Rodero and Yannick Pro, which I think begins to show some of the granularity and the detail that is coming out in the study of human interferonopathy. And a really important question is what the mechanisms are and where we can intervene. Can we potentially intervene upstream? Can we understand which nucleic acids are accumulating? Can we understand more about the sensors which they're activating and interfere with those pathways? But also, can we understand the downstream mechanisms? Can we understand the cell types that are particularly vulnerable to very high interferon levels? And we often see diagrams like this which show events which can lead to increased interferon alpha production and then downstream effects of interferon through the type 1 interferon receptor. But we often know very little about which cells are actually producing the interferon, which cells are vulnerable. And understanding that is, I think, going to be extremely important as therapeutic options become increasingly considered and available to try and optimize treatments in both interferonopathies and also other human brain diseases which I'll come on to, which are associated with very high interferon levels. And to put this in a fairly crude way, in AGS, then neuropathological studies and IPS-based studies have shown that there is definitely increased intracerebral synthesis of type 1 interferon. The exact cellular source is not known, but there is some evidence, for example, that astrocytes are producing potentially neurotoxic levels of type 1 interferon. And that means that this can be modeled well by a transgenic overexpression approach. This is a model developed with which was developed in the University of Sydney by Ian Campbell and Marcus Hoffer. We're developing a more sophisticated way of targeting interferon overexpression, but this model has worked very well for a long period of time of GFAP-driven overexpression of type 1 interferon, which leads to overexpression at either low levels in one strain and very high levels in another strain in mice. So there is recapitulates this important part of the phenotype, which is brain-driven overexpression. And we see within the brains of these mice a lot of similarities with AGS pathology. We see diffuse brain disease, we see prominent small vessel disease and microangiopathy and intracranial calcifications. And this is also potentially a very interesting model to try and identify which cell types within the brain are particularly vulnerable to very high levels of interferon and chronic interferon alpha overexpression, such as one might see in a cardiac UTR syndrome. So we've been taking with Marcus and Ian a single-cell approach, looking at the brains of these mice, and we see that actually the cells which have the highest levels of IFNAR1 expression and also IFNAR2 expression, and so have the machinery really to recognize interferon, are endothelial cells and microglia. And that may not be a huge surprise that both those cell types are particularly well constructed to respond to interferon signaling. But when we look, this is a UMAP plot here, when we have a look at changes in the transcriptomes using a single-cell RNA-seq approach, we see that, for example, in astrocytes, the wild-type mice, which are here in orange, do not differ hugely from the mice which overexpress interferon in the brain. There's not a huge transcriptional shift in astrocytes, but we do see very significant shifts in both the endothelial cells and the microglia. And so these are clearly, there's emerging evidence that these are cell types which are particularly responsive to interferon signaling and may be particularly vulnerable to disease. And when we look at the small vessels, this is a casting, a microvascular cast approach of these mice. We see that in contrast to the very regular structures of the capillaries, mice which overexpress interferon alpha in the brain have really very abnormal structured microvessels. So they have, for example, microaneurysms and changes in the vessel caliber, which are seen diffusely throughout the brain. So it can clearly, interferon alpha can clearly be toxic to small blood vessels, and this is a theme I'll come back to a little bit later on. But we're now getting an idea of where we can target therapeutic approaches, whether it's upstream at the fundamental cellular origins of this innate immune activation and interferon activation, for example, with the production of immunostimulatory nucleic acids, or whether it's more downstream targeted at circulating interferons or the receptor. We're beginning to get an idea of what cell types and where those receptors are that are particularly important to target. So I wanted to finish just talking a little bit about complex autoimmunity, which are mainly the patients that I see in my clinical practice, because I see quite a lot of patients who have lupus, and type 1 interferon has been a very important, thought to be an important mechanism in driving disease in lupus and also potentially contributing to brain disease in lupus. Lupus is a relatively common autoimmune disease, and we know that about 80% of patients with lupus have very high or have detectable interferon signatures, and we also know that there are genetic risk factors in terms of rare variants in TREX1 and RNase H2, which can confer risk for both lupus and neurolupus. And one of the difficulties in studying interferon alpha has always been detection of what is an extremely potent low abundance protein. And so single molecule approaches here, and these are part of efforts from Yannick Crow in terms of developing interferon biomarkers for use for monitoring disease activity, which have been hugely important in terms of setting the structures up for clinical trials of interferon dysregulation disorders, and also work with Dara Duffy at the Institut Pasteur, using antibodies derived from patients with APOSED with extremely high affinity for interferon alpha, and using the single molecule ELISA, digital ELISA platform, to reduce the level of detection by 3 logs. So we can now use this digital ELISA to detect interferon alpha at levels that is exerting its physiological effect. And so these are patients who attend my clinic here with lupus in the middle, and you can see that they have very significantly raised interferon alpha compared to healthy controls, but not as much as children who have AGS and genetic interferonopathies. But this also allows us the opportunity of being able to measure interferon alpha to explore disease associations in lupus. And this is extremely important now that drugs like anifrolumab, which target the interferon alpha receptor, have passed phase 3 trials, one of the two of the ongoing, of the completed phase 3 trials in lupus. It's really important that we, in very heterogeneous diseases such as lupus, identify what the specific disease associations that might be targeted by interferon blockade are. And we see in these patients that high interferon levels are associated with hypocomplementemia, but also importantly with autoantibody production. We see a dose-dependent effect in patients with lupus and patients with very high levels of circulating type 1 interferon produce very high levels of autoantibodies. And that is, there is of course a question about cause and effect there, whether that is just association. But an important way of addressing it is also to see whether interferon itself can cause the production of autoantibodies. So this is, I think, an interesting observation from our clinic. I run a clinic with colleagues in Edinburgh looking at a condition called neuromyelitis optica, which is an autoimmune disease of the spinal cord and optic nerves caused by anti-glial antibodies, particularly against aquaporin 4, which is a water channel expressed in astrocytes. And these patients often have a very distinctive phenotype. They have very high, and the presence of aquaporin 4 antibodies is very specific for this condition. And we, in fairly quick succession, had 4 referrals to that clinic, one of whom was a patient in the lupus study who had extremely high levels of interferon, who developed a cervical transverse myelitis associated with aquaporin 4 levels. There's a patient with a child with an interferonopathy due to an IFIH gain-of-function mutation who developed at the age of 3 a transverse myelitis with aquaporin 4 antibodies. And that then leads us to this question as to whether this is cause and effect. But equally within the clinic, we saw 2 patients who were being treated with recombinant interferon alpha therapy, which we know from clinical trials is known to induce autoantibodies. It's less clear that it can induce autoantibodies against neuronal and glial antigens. We had 2 patients, one of whom had been treated with systemic with interferon for systemic mastocytosis, the other for hepatitis, who both in the context of interferon alpha therapy developed transverse myelitis. And then using this as a safety signal, we then went and looked into global drug safety databases such as that held in the Uppsala Monitoring Center, and identified a link between exogenous, as it were, iatrogenic interferon and the development of aquaporin 4 mediated neuroinflammation. And we also know when we look at patients with NMO who have aquaporin 4 antibodies who have received interferon, mistakenly thinking that they have multiple sclerosis, have often fared poorly. So we know that interferon alpha worsens NMO, but also can potentially induce it. And so we have this very interesting paradox in adult neuroinflammation where we see type 1, the influence of type 1 interferon can exert divergent effects in diseases which can be very similar. So we can see that type 1 interferon alpha, can worsen neuromyelitis spectrum disorder. It can even trigger neuromyelitis optica spectrum disorder. But equally, paradoxically, a disease which can look very similar, which is often misdiagnosed. So MS, a common misdiagnosis of MS is actually NMO spectrum disorder. But interferon beta has been approved as a treatment for MS since positive phase 3 clinical trials in 1998. It was one of the most widely used immunotherapies, and it definitely reduces relapse rate by about 30% and MRI disease activity by about 50% in multiple sclerosis. And so we see that 2 superficially quite similar diseases. And we don't quite understand why type 1 interferon improves multiple sclerosis. but we don't quite know how it induces neuromyelitis optica. But we can see that there is quite a nuanced effect in neuroinflammatory diseases in non-genetic adult disease. And leading on from these observations, we also have, this is a patient here on the left who had multiple sclerosis for many years, had been being treated with type 1 interferon, which was thought for many years to be a very safe drug. It still is a fairly safe drug. And this is a patient who presented to the intensive care unit with an encephalopathy and evolving renal failure. And these arrows are pointing to not MS lesions. This is a diffusion-weighted scan which shows multiple small vessel infarcts. And this is a renal biopsy from the same patient showing a microaneurysm in their kidney biopsy, which is very similar to the microaneurysms we see in these interferon overexpressing mice. And we were able to identify as a severe side effect of type 1 interferon therapies this entity of interferon beta associated microangiopathy. And this is also seen in type 1 in interferon alpha therapies. And this is a small vessel disease that can affect the microvasculature of the brain, the kidney, of the skin, and of the heart, that can occur in up to 1 in 10,000 patients treated with type 1 interferon. And this pathology is also very similar to some of the neuropathology that we see in post-mortem specimens of individuals with AGS as well. So there is a lot of nuance in the beneficial or detrimental effects of type 1 interferon, both as a therapy but also as something which can induce severe side effects. And we're getting better descriptions and a better understanding of the spectrum of disease which it can both treat and cause. So in summary, immunity in the brain is complex. It's challenging to study in humans. There's a lot of also cellular complexity to it as to where interferon arises and where it has its effects. The type 1 interferon response is really critical for cerebral immune homeostasis. Underactivity predisposes to CNS infection. I'm sure we'll hear a lot more about that later. But equally, overactivity can cause neuroinflammation. But in adult neuroinflammation, we also see we see some complexity in the fact that it can be both beneficial and detrimental in terms of outcomes for adult neuroinflammatory disease. So I'd just like to thank members of my lab and my collaborators in Edinburgh, and I look forward to answering your questions in the Q&A session later. Thank you.
Hello, everyone. It's a great pleasure to speak at this ESID meeting. So firstly, thanks for the organizers for the invitation, and thanks to all of you for attending. So my topic will be about primary immune deficiencies in children who suffers from viral encephalitis. The mechanisms underlying such conditions has been shown in the past by us or others to be interferon related or not related. So I will give a brief overview of what we have learned in the past until 2018. That was the last ESID meeting. And then I will give you two examples of two news stories that are papers published in 2019 or in press. So firstly, a brief background for about viral encephalitis. So viral encephalitis, we know that it is a rare and severe acute viral infection of the central nervous system. However, if we take viral encephalitis caused by all different viruses together, it is actually a relatively common disease because the accumulated prevalence is about 1 person in 1,000. Many viruses can cause viral encephalitis. And so about 30% of the acute viral encephalitis have a known viral origin. At least 20 viruses can be causal. Some of them specifically infect the central nervous system, like Japanese encephalitis virus, poliovirus, and the rabies virus. Some others, they will infect multiple organs and will specifically lead to viral encephalitis only in some patients, like herpes simplex virus, influenza virus, or enterovirus. So interestingly, other than the causative virus, the disease pathogenesis really was unclear. And an interesting feature about viral encephalitis was that most of the patients, they are otherwise healthy, other than their viral encephalitis cause, means that they are not susceptible to any other severe infectious diseases, and they are not even susceptible to other severe infections caused by the same virus. And also another interesting feature is that although putting together viral encephalitis is a relatively common disease, however, if we take these viruses one by one, and then we can say that the encephalitis caused by a specific virus is very rare. We all know well, for example, examples like HSV1, influenza viruses, they infect almost everyone. However, viral encephalitis caused by these viruses are extremely rare. So in our lab, we asked a question about 15 years ago that was there or not a role of host genetic factors, in particular monogenic genetic factors underlying this condition. So I joined the field of primary immune deficiency about 15 years ago. So I'm sure that many of you know much better than me about the classical primary immune deficiency. And the viral infection of the central nervous system actually have been seen in the past in these different classical immune deficiencies. A good example is the enterovirus infection in XLA. And other examples are some case reports about viral encephalitis in DOC2 or DOC8 deficient patients. These are two classical combined immune deficiency. However, these classical primary immune deficiencies, some patients, they suffer from viral infection of the central nervous system, viral meningitis, or viral encephalitis. And in addition to that, these patients, they are also susceptible to many other infectious diseases like bacteria or fungal. And also, these conditions, we all know that they are rare, and familial cases often have been observed, and multiple infections, recurrent infections occurs to the same patient. And many times these infectious pathogens, they are opportunistic. So compared to these, most of the viral encephalitis, they are sporadic. Means that they happens in people who are not susceptible to any other infectious diseases, as I just said. And so familial cases are rare, and recurrence is also rare. And really putting together viral encephalitis is a relatively common condition in otherwise healthy people. So then, in our lab, we decided to study these isolated viral encephalitis in otherwise healthy children starting from 2006. And the first disease that we studied was herpes simplex virus 1 encephalitis. That is the most common viral encephalitis in the Western world and perhaps also all over the world. HSV1 is a neurotropic virus. It infects more than 80% of the young adults worldwide. It typically causes asymptomatic or benign infections. The virus can reach the central nervous system via olfactory and the trigeminal nerves, then cause encephalitis in some patients. It is rare for this virus infection because HSE affects only 1 to 2 in about 10,000 people globally. And interestingly, the peak of incidence of HSE is between 3 months and 6 years of age, and 1/3 of the HSE cases are therefore corresponded to primary HSV-1 infection. So given these interesting features of HSV-1 encephalitis, we decided that we would take HSV-1 encephalitis as a modeling disease to start a dissection of central nervous system specific primary immune deficiency, possibly underlying viral encephalitis in children. And so in the lab, we have observed in the past that one complete deficient patient and another NEMO deficient patient, they have suffered from HSV-1 encephalitis in addition to their mycobacterial diseases. So STAT1 and NEMO are 2 molecules which controls the production or the response to interferon alpha, beta, and the lambda. And then we firstly hypothesized that type 1 or 3 interferon might play an important role in controlling HSV-1 infection in the central nervous system. And this led to the discoveries of gene defect in the toll-like receptor 3 and interferon production pathways that listed in this table in brief. And also, lastly, a DBR1 deficiency that is not necessarily related to interferon system. So other than DBR1, all the 6 genes that are listed in this table, we can see here at the left side, from the top to the bottom, we have toll-like receptor 3, UNC93B, TRIF, TRAF3, TBK1, and ERAF3. These are all key molecules of the toll-like receptor 3 pathway that controls the interferon alpha, beta, and the lambda production upon a virus stimulation. So we have found the heterozygous or homozygous or compound heterozygous mutations in all these different genes. And then using the patient cells, we have shown that dermal fibroblast cells, they have a toll-like receptor 3 dependent response to poly-C. As I show as an example here that upon poly-C stimulation, and in fibroblast cells from patients with toll-like receptor 3 or UNC93B deficiency, the response was completely abolished as compared to healthy control cells. And then at the right bottom panel, we can see that also upon HSV-1 infection, the production of interferon was also impaired in cells from patients with toll-like receptor 3 pathway deficiencies. Then we also studied PBMCs from our patients just as a comparison to fibroblast cells. What we have seen is that in contrast to fibroblast cells, PBMC seems to have a TOL3 independent response to poly-C, that is a double-stranded synthesized double-stranded RNA that mimics byproducts during virus infection. And so these PBMCs from patients or family members with TOL3 pathway deficiencies, they responded well to poly-C and to all the viruses that we have tested. So we explained this by the fact that in different cell types, and so maybe in blood cells, the other sensors like of double-stranded RNA like RIG-I, MDA5, or other interferon-inducing sensors like CGAS, they can compensate the deficiency of TOL3 in these cells and then can induce interferon production upon virus infection. And this can at least partly explain why patients with toll-like receptor 3 pathway deficiencies, they do not suffer from any other severe infectious diseases. And then to go one step further, we studied cells differentiated from the iPS cells of our patients in order to dissect a central nervous system specific cell intrinsic immunity to virus. So we differentiated iPSCs to neuron stem cells, to central nervous system cortical neurons, to oligodendrocytes, to astrocytes, and to trigeminal ganglion neurons. And so we firstly see that as compared to the neuron stem cells, astrocytes, or trigeminal ganglion neurons, central system cortical neurons and oligodendrocytes from the same healthy control, they are able to control HSV-1 infection very efficiently. And however, these two cell types from our patients with TOL3 pathway deficiencies, they seem to lose this resistance to HSV-1 infection, as we can see from the images on the right top panel and the graph at the right bottom panel. HSV-1 infection was shown as these white dots or elevated GFP values. And so that virus infection phenotype in TOL3 pathway deficient patient neurons can be further rescued by pretreatment of interferon alpha or beta. So these experiments strongly suggested that central nervous system cortical neurons, they do have a cell intrinsic TOL3 dependent antiviral mechanism that is essential in the control of HSV-1 in the central nervous system. And this may be related to the disease pathogenesis in those children suffer from forebrain HSE. And then with this, we further asked a question is that as Toll-like receptor 3 is the inducer of type 1 and type 3 interferon. So among these different subtypes of interference, what can be the most important for control of HSV-1 infection in the central nervous system? So with this, I'm going to show a new story that has been carried on by a PhD student whose name is Paul Basta, and the paper is in press right now. So we received a consanguineous family from Israel from Dr. Ruiz. And in this family, we can see that patient 1 suffered from HSE twice. And then in his family, patient 2 suffered from 2 episodes of herpes meningitis. And the patient 3 was died of vaccination of measles. So in this family from patient 1 by whole exome sequencing, and then we figured that these patients carries a large genomic deletion in the gene of IFNAR1, interferon alpha receptor 1. And this deletion will remove part of the intron 10 and most of the exon 11 of interferon alpha receptor 1. And this leads to a truncated interferon alpha receptor 1 protein. And this truncated interferon alpha 1 protein actually was expressed at the cell surface, as we can see here from the FACS data that in patient 2 fibroblast cells, cell surface expression of IFNAR1 was there. And from the Western blot results, we can see that this protein, however, was truncated because it has a smaller size. And this truncated protein, however, even though it's expressed at the cell surface, it can still bind to interferon, but it cannot bind to the downstream kinases like TYK2, and then the patient cells completely lost responsiveness to interferon alpha 2b, as we can see at the left bottom panel. These are RNA sequencing data. And in contrast, the response to interferon gamma was intact in the cells from the same patients. And this loss of response to interferon alpha or beta was related to an increased HSV-1 replication type from the cells of P2, and with and without interferon alpha treatment. So this is briefly the story. Paul is going to talk about the story in another session, so I'm not going to more details. And then I'm going to introduce another new story. So for this story, actually we asked the question then, right now we know that toll-like receptor 3 and type 1 interferon that can be critical in the control of HSV1 infection in the central nervous system. So however, only about 10% of the patients that has been studied in our lab carries mutations in these genes. What about the other patients? Do they also suffer from inborn errors of immunity? And was that related to interferon or not? And then with this question, we studied the patients by whole exome sequencing, and very surprisingly, we found the mutations in a gene called as SNO31 that actually we have no clue it is related or not to TOL3 or interferon. The story was started by cohort level analysis of the whole exome sequencing data. We compared our HSE exomes versus controls. So we have about 300 HSE exomes, and then we compare with about 2,000 controls. And then we searched for genes that are mutated in more than one HSE children, and the mutation event is enriched in the HSE cohort as compared to the controls. And then the gene ranked as top number 1 was SNO31. So we found the heterozygous mutations in SNO31 in 5 patients from 5 different countries. And 2 of the mutations are recurrent, and the other 3 mutations each affect 1 patient. We can see that SNO31 is located in an intron very close to an exome of a gene called as TPT1. And other than this, what did we know about SNO31? If we search at PubMed at that time when we discovered the mutations, actually nothing was found, except that there was a prediction that SNO31 might bind to the 218 site of 18S ribosomal RNA and the 3713 site of 20S ribosomal RNA, and will guide their pseudourination. That was all we know about SNO31. However, we still decided to study this gene because 5 patients mutated in this gene, so it's important to make sure that if this gene is related to the disease or not. And then, so firstly, we did was to look at population genetic level. We confirmed that by looking at all the small non-coding RNA coding genes, SNO31 is among the top conserved genes in human populations that we can see here at the left panel, that SNO31 has a very high GERP score. Higher GERP score means that a higher conserved the gene is. And as a control, a gene called as SNORD118 that is related to autosomal recessive neurological condition in other patients has a much lower GERP score. And also, again, at population level, if we look at the Genome AD database that I show here at the right bottom panel, we can see that not many variations in SNO31 have been observed in the general population. Again, suggests that this is a highly conserved gene. And the 4 mutations from our 5 patients, actually 2 of them are located in the loops, and 2 of them located in the stems of this SNO31. And the 2 of them affect a nucleotide that is predicted to affect the stability of this small non-coding RNA, and 2 of them are located in the functional domain of this small non-coding RNA. And so then consistent to the prediction, we can see that the variation 1 and the variation 2, they indeed will lead to the instability of SNO31 that we show here in the overexpression system at the left panel. And then if we put these 4 mutations into the general population, we can also see that the variations of SNO31 that affects the stability of SNO31, they are not a lot. Again, this shows that mutations in SNO31 can be pathogenic. And then after these population genetics and biochemical studies, we studied at the cellular level, these SNO31 mutations by using firstly fibroblast cells from our patients. So firstly, we show that in control fibroblast cells, SNO31 is not an ISG because neither poly-IC nor interferon beta can induce the expression level of SNO31. And then in fibroblast cells from our patients, consistent to the overexpression system data, cells from patient 2 and 3, which carries variation 1 and 2, they had a lower SNO31 expression level as compared to healthy controls. And also, the cells from our patients, which are heterozygous for these SNO31 mutations, they are susceptible to various virus infections, including HSV1, measles virus, varicella zoster virus, VSV, and EMCV, and with only exception that might be poliovirus. These experiments suggested that SNO31 might be a virus restriction factor for some, but not for all viruses in fibroblast cells. And then as we have these IPS mediated central nervous system cell differentiation model, so we used the iPS cells from our patient cells and differentiated them to central nervous system cortical neurons. And here we can see at the top panel that SNO31 expression level was decreased in cortical neurons from patient 2, 3, and even 5, which are heterozygous for variation 1, 2, and 4. And all these patient-specific cortical neurons, they are susceptible to HSV1 infection that we can see at the left bottom panel, and interferon alpha treatment can rescue this virus phenotype in these patients' neurons that I show here at the right bottom panel. And so these data suggested that the heterozygous mutations in SNO31, they will lead to decreased SNO31 expression in the patient's neurons and related to a higher susceptibility to HSV1 infection. And so what can be the mechanism? How SNO31 can control HSV1 infection in neurons? So with this question, we generated a CRISPR-Cas9 mediated SNO31 KO neurons. They carries either homozygous or heterozygous deletions in SNO31. And these neurons, they shows decreased SNO31 expression levels. However, these neurons, they can be differentiated normally, and the host gene of SNO31, TPT1, was expressed normally, so means that SNO31 is not involved in this process. And however, then we experimentally proved that one of the 2 predicted function of SNO31 that was about the pseudounionation of 18S ribosomal RNA was confirmed by pseudo-seq experiment, because the homozygous SNO31 deleted neurons showed decreased signal. However, heterozygous SNO31 deletion does not affect this function. While either homozygous or heterozygous deletion of SNO31 led to an increased HSV1 replication levels in these cortical neurons. So this means that SNO31 controls HSV1 infection via a mechanism that is not the canonical function of this small non-coding RNA. And so then finally, we performed RNA sequencing in cortical neurons with different SNO31 mutations upon stimulation by poly-IC, by interferon alpha, or by HSV1. And then we observed that the response to poly-IC and the response to interferon alpha was completely normal in these SNO31 mutated neurons, means that the TOR3 and interferon pathway is intact. However, the immune response to HSV1 was impaired in SNO31 mutated neurons, as we can see here clearer in the right bottom panel, that in the healthy control neurons, we can see those red dots. Each dot is immune-related gene, and so many of them are highly induced in control neurons by HSV1 infection. However, many of them are missing in SNO31 mutated neurons. So these experiments suggested that SNO31 indeed is a central nervous system cortical neuron intrinsic HSV1 restriction factor that can have an antiviral impact as a constitutive antiviral, in a constitutive manner, And so then with all these, I will make a summary. So in brief, here is a table that then I added all the past known and the new discovered genes in patients with isolated viral encephalitis, either caused by HSV1 infection or caused by other viruses like influenza virus, norovirus, or varicella zoster virus. All these putting together, they suggested that viral encephalitis can be a group of central nervous system intrinsic inborn errors of immunity, at least in some children. So what we learned immunologically from these studies is that inborn errors of immunity, dependent on interferon or not, can underlie the development of HSV1 encephalitis in some children. That they are mediated by the TOR3 and interferon pathway or by SNO31 or DBL1 via unknown new mechanisms. And then if we put them into a bigger picture about all different viral encephalitis in children, and then we can see that the TOR3 and the interferon pathway is called, governed by TOR3, UNC93B, and finally IFNA1, they can be essential to control HSV1 infection in the central nervous system. And then DBL1, SNO31, and then POR3 can be critical in control of HSV1 and other viruses also in the brain. And I really hope that these studies can have not only immunological implications, but also clinical implications that we can use not only for the diagnosis of the patients and may be able to guide the treatment of the patients in the future. And I hope that also in the future, more people will join the effort to study these inborn errors of immunity underlying viral encephalitis. And so that will be really helpful to dissect either virus-specific or organ-specific immunity to different viruses in this particular organ. With this, I'd like to thank, firstly, all our collaborators, including the collaborating scientists and the collaborating physicians, and also our patients and their families. Their help are really very important, and also other funding resources. And then finally, these are our two labs, one at in the Rockefeller University, headed by Zhang Casanova, and the other is in Paris, the Imagine Institute, headed by Roland Abel. And here I listed the names of the people who have been or are involved in our studies of viral encephalitis. And I'm happy to take your questions, either now or via emails. My email address is here. Thank you very much.
Hello, my name is Raquel Ruiz García. I'm from Spain and I work in Barcelona, in the hospital clinic of Barcelona. I would like, first of all, to thank to the ESID to inviting me to this clinico-pathological discussion session to talk about these fascinating rare autoimmune diseases, which are the autoimmune encephalitis. I have nothing to disclose, and well, it's very well known, the connection between primary immune deficiencies and autoimmunity, right? And then we can say that there are two main groups of autoimmunity present in these primary immune deficiencies. So the ones that present autoimmunity at the main as the main manifestation of these diseases, like the very well-known iron deficiency, FOXP3 deficiency, and also lymphoproliferative syndromes, CTLA4 haploinsufficiency, and also STAT3 gain of function, and a large number of primary immune deficiencies, right? And there's also a group of primary immune deficiency with susceptibility to infections that has also autoimmune manifestations or inflammatory manifestations. So in this very, very nice study done by the French group, published a few years ago, these groups analyzed more than 2,000 patients with primary immune deficiency and observed that the 25% of these patients had an autoimmune or inflammatory manifestations. And this percentage comes much more higher when these patients have 50 years or are 50 years or more of age. So when they analyze the autoimmune manifestations by the clinic, so they saw that the most common manifestations are the autoimmune cytopenias and also gastrointestinal disorders. And when we look for the neurologic disorders or autoimmune neurologic disorders associated with primary immune deficiencies, we saw that only 8 of 852 manifestations are from neurologic features. So it's only 1% of the overall autoimmune manifestations. And when we see this profoundly, we see that the manifestations were myasthenia, intracranial and intraspinal granuloma, progressive multifocal leukoencephalopathy, and also encephalitis, myelitis, and encephalomyelitis, but not specified. So these are rare manifestations, but also occur in patients with primary immune deficiencies. When they analyze the relative risk of having a neurologic disorder in these patients with primary immune deficiency, it's 3 times much higher than the general population. It's not very high, but it's 3 times higher. And maybe you have seen this picture of this cartoon before in the previous talk, so I would not spend much time in this, but it's just to remember that primary immune deficiencies are very well associated with viral encephalitis of virus from the herpes viridae family. And there are a group of immune deficiencies in the signaling of the TLR3 that causes susceptibility to herpes simplex encephalitis. So these are well-characterized primary immune deficiencies and has been previously probably reviewed before. So, but the question should be or could be, is this autoimmune encephalitis linked or related with primary immune deficiencies at all? Have they anything to do? So I would like to start with some history about the autoimmune encephalitis. So this is Dr. Corsellis. Corsellis and his colleagues published in the 1960s this paper collecting a group of patients that had lung cancer and suddenly developed some memory disturbance. They suddenly develop an inability to form new memories, and they call this entity limbic encephalitis. So a few years later, when they analyzed again this patient, they saw that they have lymphocytic infiltrates in their brains, and especially in the limbic area. So this is the first time that they associated this immunologic manifestation associated with these memory problems, so linking this with an encephalitis. And there, by this time, there were another group studying the same group of patients that discovered that these patients with lung cancer and brain neurologic manifestations had also antibodies in their serum and also in their CSF that reacts with neuronal tissue. So they react with neurons on slices of García pig. This is very important because this led to other groups to investigate in this kind of diseases, and it was in the late 70s where Dr. Posner and Dr. Graus discovered that these antibodies present in this patient recognize intracellular epitopes of the neurons. So they led also to first start talking about the paraneoplastic neurological syndromes. These syndromes were characterized because patients had intracellular antibodies against neuronal epitopes, but these antibodies react with intracellular antigens. So they were they could not access or to the epitopes, or they could not directly cause a damage on the neurons. So they are not directly pathogenic antibodies. These diseases, these neuronal diseases, were always associated with tumors, sometimes lung cancer or breast cancer or ovarian cancer. And a few years later, they discovered, when they studied more profoundly the autopsies or the necropsies of these patients, that there were lymphocytic infiltrates with CD8 cytotoxic T cells attacking neurons. So these antibodies were markers of the disease, but the neuronal damage were produced by CD8 T cells. These patients have a very poor neurologic outcome after immunotherapy. They don't respond well, and this is important because we are going to see another group of diseases that recognize that will have also antibodies that recognize surface antibodies and have clinical implications. About 30 years later, Dr. Dalmau discovered another group of diseases characterized because of the patients had also antibodies, but these antibodies react with neuronal surface antigens. So these antibodies were discovered to be directly pathogenic and directly cause the disease. They do not produce a very large damage, but react with some receptors of neurotransmitters and other proteins, causing functional disruption of the neuronal synapse. So these patients had a good response to immunotherapy. All the therapies directed to eliminate or remove these antibodies are well to these patients. They respond very well, and they sometimes recover completely. And these are not necessarily associated to tumors. So if we see the cartoon here, what we see is these antibodies recognizing the surface of a protein, the surface of a neuron. And what we see in the left panels is the reactivity against rat brain tissue, where we saw the hippocamp of a rat stained in the dentae and also the superior part of the hippocamp. And also when we study cultures of live neurons, we see these antibodies reacting with the neuronal surface of these neurons. And this goes in contrast to what we have previously seen before. When we have these intracellular antibodies, they could not access to the live neuron, and there's no staining. This is very important because it has some treatment and prognosis features that we can see after that. So nowadays we recognize 17 different diseases caused by these surface neuronal antibodies. Most of these diseases recognize neurotransmitter receptors as the NMDA receptor encephalitis, and also AMPA receptor encephalitis, and also other metabotropic glutamate receptors or dopamine receptors. And these diseases are associated sometimes with tumors, but it's not mandatory. There's some diseases that have more percentage associated with tumors, but others that are not associated at all with them. This is a picture showing the different reactivity of these antibodies on the rat brain tissue. So the rat brain proteins are very similar to human proteins. So this is the methodology we use to rapidly detect these antibodies on CSF and plasma samples. And I'm going to talk a bit more about the NMDR encephalitis, because this is the best characterized encephalitis and also the most frequent. So you can see again here the reactivity on rat brain tissue and also in neurons, and also what we do to confirm the presence of these antibodies is that to transfect the human protein on a cell line and then do an immune cytochemistry to detect these antibodies on the plasma and CSF from the patients. So these antibodies against the receptor of NMDA produce an internalization of these receptors, so that led to a reduced density on the neuronal surface of the receptor, disrupting the synapse or the signalings of the synapse produced with glutamate and glycine. This is a very nice graphic that describes the complexity of the clinical symptoms that these patients have. So they usually start with a viral-like prodrome and then rapidly evolve to some psychiatric manifestations, psychosis, agitation. They also have hallucinations or mania. And if they are not treated properly with immunosuppressants, they rapidly evolve to catatonia, sometimes coma, and started with dyskinesias of abnormal movements. If they are properly treated, they rapidly pass these symptoms that are more severe, but there's a period where these patients continue having this memory deficit, also with impulsivity, and these are the symptoms that last more in the time over the recovery. And this very nice study that studied a cohort of 577 patients with NMDA encephalitis, what we saw that maybe is important for you, because I guess most of you are pediatricians, is that when they separate the patients in gender and also in age, what we see is that the younger the patients, the less differences between gender, and also the less association with tumor, in this case with ovarian teratoma. So maybe this is important to detect or to think about of NMDA encephalitis. Well, the MRI is not always abnormal, but maybe helps to distinguish between other diseases, and also there are some features that are common in the CSF. What triggers this autoimmune encephalitis? Well, sometimes could be an ovarian teratoma. This kind of benign tumors could have neurons and also express antigens expressed by neurons. So there could be an immunization against these neuronal antigens. But also, it is known that the herpes encephalitis could produce this damage to neurons and expose some antigens or neuronal antigens and produce an immunization against neuronal epitopes. This is important because in this study, there's a very nice study performed by Dr. Thais Armangué, where she studied a prospective cohort of patients with herpes simplex encephalitis, and studied then do a follow-up of this patient, and discovered that 27% of these patients that has an herpes simplex encephalitis develop an autoimmune encephalitis with neuronal surface antibody. So most of these patients had an NMDA encephalitis, but also there were another group of patients that had other antibodies that react with the neuron surface, demonstrated by live neuron cultures, but of these antibodies react with unknown antigens. The median of the develop of the autoimmune encephalitis were 2 months, and the symptoms these patients present were age-dependent. Children present more frequently dyskinesia, so movement disorders, and the adults present more frequently behavioral disorders. The prompt diagnosis led to a better outcome because this patient could be treated with immune suppressant and have a better outcome. There's also other groups of encephalitis, autoimmune encephalitis. I would like to talk about the GABA-A receptor encephalitis because it's frequent in children and clinically, it's very important that these patients had a lot of epileptic seizures and also status epilepticus, but they respond very well to immune therapy. So to detect these antibodies could be very important to start a treatment soon. They also have these characteristic MRIs with multifocal lesions. And also I would like to talk a bit about the encephalitis produced by antibodies against the myelin of oligodendrocyte glycoprotein. So these antibodies were first described in association with the myelin-19 syndrome, such as neuromyelitis optica. But in this study performed by our group, also led by the Dr. Armanguer and Dr. Dalmau, they identified that the 7% of patients that have an encephalitis different from acute disseminated encephalitis had antibodies against MOG. And this is very important because the identification of these disorders could have clinical and prognostic implications. So it's something to have in mind that these antibodies could be present not only in the myelin-19 syndromes, but also in autoimmune encephalitis. So I would like to talk about this case because I think this is the case that could connect the primary immune deficiencies and also autoimmune encephalitis. So this is a case that was published last year of a 6-year-old girl that had as relevant a family history of herpetic keratitis. Her grandfather had herpetic keratitis. So she presented with acute onset headache, also fever, and decrease of consciousness and hemiparesis. The MRI showed bilateral hemorrhagic temporal lesion that led us to an herpes simplex encephalitis suspicion that was posteriorly confirmed. She was treated with acyclovir and discharged after 21 days of treatment. 5 months later, she was readmitted again at the hospital with severe headache and decrease of consciousness. And also the MRI showed these bilateral hemorrhagic temporal lesions, suspecting a reactivation of the herpes simplex encephalitis or a new episode of herpes simplex encephalitis that was confirmed. She was treated again with acyclovir and also oral acyclovir, and we performed a genetic study that showed a novel heterozygous variant in the TLR3 gene. 1 year later, and 17 months after the first encephalitis episode, she started with behavioral changes, paranoid and aggressivity. And the CSF showed elevated proteins and pleocytosis, but all the tested to search for herpes simplex virus were negative. So this patient had a novel heterozygous mutation on the TLR3 gene. Her mother also has the mutation, and we couldn't study her grandfather, which was the one that had herpetic keratitis, because he was deceased at that moment. So this defect on this gene produced a negative impact on the TLR3 signaling, and this graphic shows the production of interferon beta of the fibroblast of the patient after the stimulation with poly-IC, and we saw there's no production of interferon beta in contrast to the to fibroblasts of a healthy control. So this third episode of neurologic manifestation led us to investigate whether if these patients had any antibody against neuronal surface antigens. So this was confirmed by the immunohistochemistry assays on rat brain tissue, and also by immune cytochemistry in live neuronal cultures, but it was negative for the known antigens, for the known antibodies against known antigens like NMDA receptor, GABA-A receptor, etc. She had a neurological improvement after rituximab therapy, and 4 months later there were no signs of the presence of neuronal surface antibodies. We retested it again, and the antibodies had been removed or disappeared after the immune therapy. So in summary, or the key messages of this lecture should be that the autoimmune neurologic manifestations are very, very rare in patients with primary immune deficiencies, that patients with herpes simplex encephalitis could have neurologic relapses of an autoimmune etiology, and I guess most of you are pediatricians that also are infectologists, so this is important for you to remind that there is antibodies that could be detected, and it's recommended to search for neuronal surface antibodies in this type of patients because they are treatment responsive to immune therapy. And also that patients with inborn errors of the TLR3 pathway should be carefully followed for potential relapses of not only, again, herpes simplex encephalitis, but also for autoimmune encephalitis produced by antibodies against neuronal surface antigens. So I would like to thank you, and also thank Dr. Dalmau and Dr. Armangué from my institution that taught me everything I know about autoimmune encephalitis, and also to you for listening to me.